Hello, welcome to another video on molecular biology and biotechnology with Lucy. Today we'll be talking about DNA replication. If it is your first time to watch these tutorials, feel welcome. Remember to subscribe to this channel. If you are following us and have been following us, thank you. For all of us, remember to like this video even before you watch it because there is great content in it that is going to help us to all learn together. And remember also to share with your friends, to comment, and just keep sharing and sharing and sharing. So today we'll be talking about DNA replication. And this is a process by which DNA makes a copy of itself so that it is transmitted from one generation to the next once we have the cell division. And it usually will occur in the S phase of the cell cycle. So this is the cell cycle process. Usually there will be what we call cell division, then there will be G1 phase, cell growth, there will be S phase where we have DNA replication, and then we'll have again the G2 phase, now that prepares again for mitosis, then mitosis, and so on and so forth. So for us to have cells multiplying within our system, we also will need to multiply the DNA so that it makes a copy of itself for the next generation of cells. So how does DNA actually replicate? We usually have several hypothesis models, and hypothesis means that they need to be tested or they were tested so that we can find out which is the correct model of replication. So there are three hypotheses. The first one is conservative replication, we have semi-conservative replication, and we have dispersive replication. Conservative replication implies that parental double helix DNA will remain intact with successful or subsequent copy processes. And that means that the daughter double helix is going to have both strands that have been newly synthesized. And here we have a pictorial of the same. So this is your parent DNA double helix from the parent cell. So for the first replication process, we are saying that both of the strands are going to be newly synthesized. So we are going to have new strands that will make this double helix and totally new strands again for the other double helix. And then with subsequent, now the second replication again, we are going to have new synthesis, new synthesis, new synthesis, and new synthesis of each of now the previous parent in the first replication. Semi-conservative replication, which is a model that was hypothesized by Watson and Crick, usually implies that one strand from parent is found in each of the new strand. So if this is our parent cell, then each of the new strands are going to consist of the parent strand and now a new strand, and so on and so forth, even in the subsequent or second replication. In dispersive replication, we have that at the end, both strands of the helix are going to contain the original and the newly synthesized, some sort of a mixture or mosaic. And you can see the mosaic and mixture in these two, and even in the second replication process. So those are the three hypothesized models: conservative, semi-conservative, and dispersive replication. An experiment was done by Meselson-Stahl to define and identify which is the mode of replication. And this was done by using isotopes of nitrogen, that is isotope 15 and 14. And in this case, what happened is that bacterial cells were added into a medium containing nitrogen 15. And this is now the original DNA. Okay, the first media that had nitrogen 15, of course, consisted of the original DNA. and therefore the DNA from the bacteria that had been grown in this medium appeared as a single band that indicates the heavy isotope of nitrogen 15. Then there was spinning that occurred, and then we transferred the medium here into another medium of now nitrogen 14 to allow for the first replication. So after one round of replication, the DNA appeared as a single band, but one that had intermediate weight. Okay, so intermediate weight means a mixture of light and heavy. And now that means it indicated that there is a parental strand in it, and there is also a new strand in it. And then we had another round of replication by continuously adding into still nitrogen 14 and spinning. And after this second round of replication, DNA appeared as two bands, one of lightweight, okay, and another of intermediate weight. The lightweight is here and another of intermediate weight. So we can see that from the first, now the parents who are generated in the first round of replication, once they formed a new strand, remember we said that each of them, if it is semi-conservative, is going to have the parent strand and a new strand. So for this one, the one of them had the parent strand and a new strand, and then for the brownish one, a new strand and the parent strand, and so forth and so forth. So then we have two of them that have intermediate weight and two of them that have now the lightweight, which is just nitrogen 14. Then we allowed for more replication in nitrogen 14 media, and the samples, of course, taken after additional rounds of replication appeared as two bands, and these are the two bands exactly as it is in the previous second round of replication. That is a lightweight band and an intermediate band. And this indicates for us that the DNA replication in E. coli is semi-conservative, and this now represents other living organisms. So just to mention, if the for example, it was just conservative, we would only be having the heavyweight and the lightweight and no intermediate bands. Then if it was dispersive, and that means that there's a combination of original and newly synthesized, then they would all be intermediate. But we can see that from the experiment, we have both lightweight and intermediate, meaning that this is semi-conservative.